last weekend we had a family meeting of people with vascular EDS in Seattle at the University of Washington. And Lara was there, and one of our genetic counselors, Lauren Puryear, talked about Genetics 101 for the Vascular Day. And she liked it because she thought that it would be something that sort of unwinds some of the complicated aspects of genetics and made it pretty straightforward, I think. So I pilfered her slide set. And as you can see, I'm also not Lauren Puryear. I'm Peter Byers. I haven't changed at all, but so thought we would run through this. And why is genetics important? So when you hear Ehlers-Danlos syndrome being talked about, it's talked about as a member of a group of conditions that are called heritable disorders of connective tissue. So heritable is an interesting term because it means that it can be inherited, but it doesn't always have to be. Now, that doesn't always have to be means that you don't need to have an affected parent to have a genetic condition because genes change in the course of transmission from parents to child. So this is what Lauren had, and how does it work? So a little refresher on high school biology. Now it's getting to be grade school biology. I mean, they were just moving it all just way early, and kids are coming up and talking about things that we would never have imagined. A little bit about how do genetic mutations happen, reading a genetic test, and then what can I do with the genetic information. Okay. So as Lauren said, there won't be a quiz. She didn't give a quiz, but I hold on to the option. So every cell in your body, with the exception of red cells, which have disposed of their nucleus, have 2 copies of each gene, also with the exception of sperm and eggs, which have only 1 because they have to unite again and get the copy number back. So this is a diagram of a cell. My pointer works on this one. Okay. So here's the outer membrane of the cell. Here's the nucleus of the cell. Each of these little crosses are chromosomes, and chromosomes are long, continuous strands of DNA. And then there are organelles in the cell. Proteins exist throughout the cell, in the membranes that separate things, and also actually on chromosomes. Chromosomes are long, continuous strands of DNA, and they're very, very tightly wrapped with a mechanism so that you can contract about a yard of DNA in its strands here into something that you can't even see. It's smaller than the tip of a pencil. So there's a very complex winding thing. And any of you ever fly those little balsa airplanes that have a rubber band propeller? Remember that? So you wind it up. You start with these 2 strands because it's hooked on, and you start winding the propeller because you want to make it go. And you get it, it winds and it winds. And then you get to a certain point, and it kind of goes, and now you begin to get the next level of winding. So you get that thing in there where you've got a knob at one end, and then you get another knob and another knob and another knob. That's very much like what happens to DNA in a cell. That's how it winds. It uses things to do that. So, and the DNA is represented as 2 strands, and it's made up of 4 building blocks, or nucleotides, which are your friends, A, C, G, and T. And those letters stand for 4 different building blocks. And in the 2 strands of DNA, like this, wherever there is I don't think those letters there's a letter, but wherever there is a C on the other strand, there will be a G. So you can see that here. And where there's an A, there's always a T. So that's the complementarity, and that's how DNA gets back together. It's also how it copies itself. Because if you break these strands apart and then allow it to read this one, it'll make that one. So this is one of the fundamental understandings that in the paper that described the structure to begin this things. Crick and Watson said, we are not unaware of the implications of this for replication. Very, subtly understated. So you can have a variant here, and so if there's a normal in that sense, a C that's sitting at that position, and you get a change in the C, then it'll repair and put a G on the other side, whereas originally it's a C and a T. So the genes that we have about 20,000 genes that are encoded. You've heard a couple of dozen genes that are involved in making forms of Ehlers-Danlos syndrome. And the COL3A1 genes for vascular Ehlers-Danlos sit on this arm of chromosome 2, and right next to them are the COL5A2 gene. It's the next gene away. And in some instances, we have people who have deletions of both the COL3A1 and the COL5A2 gene. But the phenotype looks like that, is the clinical condition, looks much more like just plain old vascular EDS, or vascular EDS when it's missing. So mutations can occur there on the one copy. And then the form of transmission for most of the common forms of EDS, so for hypermobile EDS, we think it's dominant. For vascular EDS, it's dominant. For the classical form, it's dominant. It's called autosomal dominant inheritance. So autosome means those chromosomes that are not the sex chromosomes, the chromosomes that are not related to producing gender. If you look down here, you can tell what kind of person, well, you can't tell what kind of person, but you can tell what gender of person. You have no idea what she's like. But this person had 2 X chromosomes, and that makes it female. They didn't use a Y chromosome because a Y chromosome is really squiddly small. It's about that big. And it has, most of it, what it has is information for making a person a guy person. So you see that there are 22 pairs, and then women have 2 X's and a guy has an X and a Y. So, these 22, so from here down to here, down to here, down to here, including 22, are the autosomes, and these are the sex chromosomes, the X and Y. So when it's talking about autosomal dominant, it means that there is one change on a copy of one autosome. So you could use here, for example, chromosome 2, and here's the COL3A1 gene, and one copy has been changed and one is not changed. And the other partner has 2 unchanged chromosomes. So this person here, this is, I don't know why they're this way. This is somebody who made these things like Legos. So this is a Lego person with Lego hair. And you can see from the long hair, this is obviously a woman. Right? So when we have children, we transmit only one copy. It can be either this copy, or for the guy, this copy or this copy. And you can imagine if we transmit more than one, which happens. So for example, Down syndrome is a condition where there are 3 21 chromosomes, 2 of which came usually from the mom, and then the other came from the dad. So, and that's a very complex condition, and as the chromosomes get bigger, trisomies and even monosomies, that means trisomies means 3 copies, monosomy means only 1 copy, are usually not viable. So there's just a small range in which you can have more than 1 copy. But the whole system is designed for you to transmit 1 copy each and then have children. So in that situation, you can see here is a girl who has an altered copy of the gene and has the dark coloration so that on a pedigree, on a picture of the family, often the affected people are darkened. So this is an affected person who received the affected copy of the gene from her mom. Here's an affected guy who received the affected copy from his mom. And then these are 2 unaffected children who received an unaffected copy from each parent. So the each child has a 50% chance in this situation of inheriting an autosomal dominant trait or condition. And you can, by chance, you can imagine, if it's 1 out of 2 here, we say chance has no memory, so that it doesn't matter what happened this time, you start new. So you can, by chance, have 4 affected, or you can have 4 unaffected children in this setting. So you can't always tell what the story is about it. And that's by chance. That is, your body, it doesn't pick out the altered copy or the unaltered copy to transmit. It does it by chance. How do new mutations occur? So in some families, and for many people, so about half the people that we've identified with vascular EDS have unaffected parents. So, what's the explanation? Well, could it require 2 copies? Was the milkman affected, or the postman? I'm just not. For those of you in the room who have parents and are affected, I'm not talking about your families. Or, much more likely, is that there's been transmission of an altered copy of the gene, but that occurred during the process of making copies of the DNA in the course of making sperm or making eggs. And we call that a de novo mutation. And we have every copy of our genome, of which we have 2, has 3 billion building blocks. Every time a cell divides, it has to replicate or copy those 3 billion twice. So the chances of making an error are small because the machinery is designed so that it's a very, very small rate of error making. But it's not 0. And it's not 0 in many senses because if it were 0, we would all still be bacteria. That is, we would not have had the chance of having changes occur over hundreds of millions and billions of years to the genetic material that makes the difference between organisms that all share a common form of DNA, but there have been changes over the time that does it. So that part of evolution is dependent on having an error-prone or a very, very low error rate of replication. So mutations occur here. You can see here's a copy of here's one of the chromosomes and one of the copies of the DNA. It duplicates and they go on to the separate cells. The new variant occurs in that case, so you can see it. Here's the new variant. It gets transmitted to a daughter cell, and then after that, it's all the daughter cells of this cell will have that variant there. So that's how you get them. Say that there are different kinds of changes in genes, and the genes really are the instructions for making proteins. And this is a model airplane protein. And some of them will tell the DNA to stop making a protein, and others of them will tell it to make a protein that has errors in it or has changes in it. And depending on how that works, the clinical picture in you may be different. Now, I didn't go over here recessive inheritance, but each parent is a carrier for an altered copy of the gene, but is unaffected. And that's why we call it carrier and not being affected. So a number of the forms, of EDS are recessively inherited. The dermatosparaxis form, the kyphoscoliotic forms are both inherited. And some of the forms that Anne DePape talked about this morning are inherited as recessive, which means each parent is a carrier, and the affected person has 2 altered copies of the gene. So when a genetic sample, when you're tested, these days, almost everybody is tested by sending blood. When we used to do testing initially, we would get skin biopsies from people, and we would grow the cells, and we would look at the proteins that were being made. And now we look only for practical purposes at the gene. So Lauren had picked this out from a genetic test. This is the BRCA1, sometimes called BRCA gene, which is breast cancer, BR for breast, CA for cancer 1. So this is the first of the breast cancer genes that was identified. And then here's what it says. So this has a bunch of information. It has the chromosome on which it resides, which is chromosome 17. It has the position on the chromosome beginning from the short arm all the way at the top of the way that the chromosomes are extended. And it also says which building block is changed to what building block. So that's C to an A. And then there's a one-letter code for amino acids. There are 20 amino acids. And this says that there's a glutamic acid that is changed to there's an X there, which means a stop codon. So here's how it's written out. This is in the gene, but we usually read them in terms of the gene is, in order to make a protein, has to be made into an RNA copy, which is short and has all the coding sequence together, and it's out, the protein coding doesn't use the DNA on a chromosome directly, but it has to make a copy of it in order to make a protein. So it takes it out and it makes an RNA copy. And so that RNA copy is called the coding copy. So that's what the C period here is. And so at position 3,748 in the coding copy, there was a G that was changed to a T. So that's interesting because here it's a C to an A. So that G is a C, that T is an A. So remember C's and G's, C's and T's always, or C's and G's always interact, and A's and T's always interact. So the gene name of it here is reading the other strand. I'm not quite sure why they did it that way. That's not how it would ordinarily be reported. It would be reported usually like this. And what it means is that at the protein level, so P, the glutamic acid at position 1250 in the gene, is changed to, instead of 3 nucleotides that say another amino acid, it's changed to one that says stop making the protein here. And actually what they do is that makes the RNA very unstable, so you only make half the amount. So the gene, that's the coding sequence. That heterozygous means that it's got one altered copy. And then it's classified as being something that's pathogenic. That is, this is causing the predisposition for breast cancer. And that's known first because it dramatically interferes with the function of the protein. And the second is that there are data accumulated over time about people who have these changes, or in families. So one way to determine that this is causative or pathogenic is to look to see if there's a family history of breast cancer and if everybody in the family who has breast cancer has that copy. And if everybody above a certain age, so if everybody above 60, for example, who is potentially at risk to develop breast cancer doesn't have a copy. So that, seeing it go so that only the affected people have this copy is the part that tells you that it's altered. So this is another gene, and this is actually a report from our laboratory. And so we tell you it's a heterozygous, likely pathogenic. We think it's pathogenic, but there hasn't been anybody else, and there aren't family studies to be sure that it is. But we know that from the character and the kind of substitution it is, that this is going to be damaging. So it tells you the gene. This is a nickname for the gene on this chromosome. It's where it is in the coding sequence. This is, again, the change, the position coding sequence 2083, a G to an A. And here there's a glycine, so GLY is a glycine. That position 695 is changed to an arginine. And so that's, again, that's what that information is. You don't need to know this. You just need to know what the implications are and that it is diagnostic. So the important thing is this or this. So there's the information. Okay? So what can you do with this kind of information? So the first is you can make some predictions based on the kind of variant. So for example, the COL3A1 variants, which are associated with forms of vascular EDS, have a different effect depending on what happens. And that is, if they have a premature stop codon, that is, it says stop making the protein here, which also says this RNA is trash and we're going to throw it out. That group of people has a much milder clinical presentation. They present on average 20 years later than the people who have something that makes something wrong with the protein, and they have fewer complications. So we haven't seen pregnancy complications with them in women at all. And they usually don't have the GI complications. So you heard about the GI complications earlier this morning from both people who talked. And then if there's a substitution for an amino acid, amino acids differ in size. And so it depends somewhat on the size of the amino acid that's changed, what the outcome is or what it can be. It's a little harder to sort out sometimes for doing that, but it's the kind of information that we can get. One of the things to be very careful about this sort of information, where it says, well, this gives you information about what can be expected and where you might fit on a curve and things like that, is that the curve describes a population of people. And if you're on that curve, you're on that part of the curve where people are alive. But you cannot predict for yourself, if it says, okay, for people with this variant, the mean or the median survival time is 43.5 years, you can't say you're going to be at the 43 mark or you're going to be at the 73 mark. I mean, you just can't do that. This is not good enough. There's a lot of other things that happen that add to variation. So be very careful about that. And, if you go to the literature and you say, oh, the mean survival time in people with vascular EDS is 50-something years, this doesn't mean that's you. It means it could be, but you could differ from that. So the other information that's really important that you get from this are things about reproductive planning. So, for example, the thing that's really changed dramatically over the years is that it's now possible to do things like pre-implantation genetic diagnosis. And this involves in vitro fertilization, which means you take most of the fun out of getting pregnant, because this involves giving the woman partner drugs that induce ovulation, and not just one ovulation, but multiple ovulations. And this is not pleasant. And the guy, guys are easy. To contribute a sperm sample doesn't cost anything. You test embryos, so you take the eggs, fertilize them in the dish with the sperm, and then you let them grow for a little bit, and so they've got several cells, and you can take 1 or 2 cells from the embryo and test those cells for the presence of the altered copy of the gene from one of the parents. And then you can choose those ones that are unaffected to transfer. So it's not cheap. It's about $15,000 a time. And another is prenatal diagnosis, and that is for conditions which are very severe, and the fetus is not going to survive beyond, during the term. You can make decisions. So you can test the pregnancy early. You can test it at 10 weeks by chorionic villus sampling, that is to take a bit of the placenta, or you can take the amniotic fluid, which is done usually a little bit later, about 15 or 16 weeks. And there are now cell-free DNA systems that use maternal blood. So it's for some genetic conditions, it's possible to take that at quite early in pregnancy and test, so for example, for chromosome number. And it's likely that not too long in the future that if the mother is unaffected with, say, whatever condition it is, the father is affected and has a 50% chance to transmit, and the mother has not had a previously affected child. And that's because mothers harbor a few cells from the previous children, or can. Then you may be able to detect the altered copy of the gene in blood from the mom, because these DNA things are turned over by the fetus, and then some of the DNA gets into the maternal blood. So that's another part of the reproductive plan. So that's Genetics 101. You now know everything I know about genetics. You will be able to interpret your test results, and you'll be able to you may know more than your doctor, actually, at this point. So epigenetics refers to modification of DNA that affects the function of the genes, and that in some instances is inheritable, that sort of modification which is not of the building blocks of DNA, but of modifications of it. And in some cases that can be inherited. And it's used to turn on and turn off, and theoretically there could be alterations in regions that are susceptible to epigenetic changes that could be turned off and could do that. So the answer is yeah, it could be, and in which case if you have a person who is a carrier for a recessive disorder and the other copy is turned off epigenetically, this is what happens in cancer, actually. It's one of the cancer mechanisms of turning off the other copy of the gene, is that you get other modifications that can occur. So the answer is yes, it can. But it's more likely to be very localized as opposed to having been there right from the beginning.